Going from proteins to nucleic acids is a really nice transition. This is because remember that the amino acid sequence of polypeptide, which is so important for proper folding and functioning of the protein, is programmed by the DNA, by the nucleic acids in your cells. Typically, one gene will hold the information on how to produce a protein or a family of proteins. And so the genes, these nucleic acids, are really what hold that genetic information. Again, genes consist of DNA. DNA is one example of nucleic acids that are made out of these monomers that are called nucleotides. So in this section, we're going to talk a little bit more about nucleic acids, their structure, and their function. Now, there are two different types of nucleic acids, and both are really important in protein production in a cell. These are deoxyribonucleic acid, or DNA for short, and then ribonucleic acid, or RNA. Both of them, again, participate in this process of gene expression. So DNA is really what holds the genetic information, and the RNA helps to transfer that information to the building process that builds the actual proteins. And on the right-hand side here, we have a figure of how this process of gene expression takes place in eukaryotic cells. So remember, in eukaryotic cells, the DNA is going to be housed within the nucleus. This is where the DNA is read, and small segments of the DNA are going to be copied into molecules of RNA. This is a special kind of RNA called mRNA, which we'll talk about in a later chapter. So this mRNA holds the information for that one particular gene or segment of DNA. The mRNA will make its way out through one of the pores here of the nucleus out into the cytoplasm, and it'll find itself a polypeptide building machine, the ribosome. The ribosome will latch onto that RNA, start reading the information on the RNA, and build the polypeptide here shown in purple. Now, nucleic acids, DNA and RNA, are called polynucleotides because they are polymers of these monomers that are called nucleotides. So poly, many, nucleotide, that's the building block of a nucleic acid. Nucleotides are probably some of the most complex monomers for these macromolecules that we're currently discussing. Each nucleotide consists of three components. It's got a nitrogenous base, and there are multiple options for these. It's got a pentose sugar, so it has a monosaccharide in it. There are two options for these. And then it's going to have one or more phosphate groups, depending on whether the nucleotide has been attached to a polynucleotide or whether it's still free-floating as an individual building block. If you just take the nitrogenous base and the sugar and stick those two parts together, you have an incomplete nucleotide. We call these two components together a nucleoside. Then finally, when you add the phosphate group to the nucleoside, you have the full nucleotide. So what we're going to do next is we're going to take a closer look at the different nitrogenous bases that you can use to build nucleotides and the different pentose sugars you can use to build the nucleotides. So first, the nitrogenous bases. There are 5 of these in cells. They are grouped into 2 categories or 2 families. You've got your pyrimidines, and then you have your purines. Pyrimidines include cytosine, thymine, and uracil. Now, these 3 nitrogenous bases have a single ringed structure in terms of their carbon skeleton. Purines are a little bit different. There are 2 of these, adenine and guanine, and they have 2 fused rings as part of their carbon skeleton. This is where we also introduce some differences between DNA and RNA. Both DNA and RNA have the ability to use 4 different nitrogenous bases. So in DNA, you've got your cytosine, you've got your adenine, you've got your guanine, and then DNA chooses to use thymine as its 4th nitrogenous base. So A, T, C, and G. RNA also uses C's and A's and G's, but instead of the thymine, it chooses to use the uracil. So the 4 nitrogenous bases for RNA are C, A, G, and U. The other difference between DNA and RNA nucleotides is the pentose sugar that is used to build the nucleotides. There are 2 different sugars, one for DNA and one for RNA. So DNA is called deoxyribonucleic acid because it uses the sugar deoxyribose. RNA is called ribonucleic acid because it uses the ribose. Now, these two sugars are pentose sugars, so they are 5-carbon monosaccharides, and they are both nearly identical. The only difference is on the second carbon here in the ring. So here's the first one, here's the second one. On the second carbon, ribose has a hydroxyl group, an OH, and deoxyribose just has a hydrogen. So deoxy without oxygen, that's telling you that somewhere in there, in comparison to the ribose, the deoxyribose is missing an oxygen. Okay, and so let's build now the full nucleotide using these 3 components, the nitrogenous base, the sugar, and the phosphate group. In this block right here, you have a blown up nucleotide that here is shown as part of already a string of nucleotides or a polynucleotide. So one nucleotide contains one of the 5 different nitrogenous bases. It contains a sugar. If it's DNA, it'll be deoxyribose. If it's RNA, it'll be ribose, and then a phosphate group. Now, the way that you attach one full nucleotide to the next nucleotide is you're going to make a covalent bond between the phosphate of one nucleotide and the sugar of the other nucleotide. So we're going to make this bond right here. When we do this, we essentially create a sugar phosphate backbone. It's alternating sugar, phosphate, sugar, phosphate, sugar, phosphate, and so on and so forth. This is very similar to how polypeptides are put together, right? You have that similar backbone of all of the amino acids that are stitched together. And then for the amino acids, you have the R group hanging off to one side. Well, in the polynucleotides, yes, you have that sugar phosphate backbone. And then all the nitrogenous bases are hanging off sort of the backbone to one side. The other important thing to note here is that much like with the polypeptides where you have 2 different ends, you have a carboxyl end and you have an amino end, same thing goes for polynucleotides. We call these 2 different ends kind of like having a head and a tail, the 5' end and the 3' end. We get these names from which carbon on the sugar is closest to that end. So let me walk you through this. So if we're looking at this last nucleotide here, and here's the sugar, and the sugar carbons are numbered starting with the carbon that's closest to the nitrogenous base. So this is carbon 1, 2, 3, 4, and 5. So if we go back here, you'll notice we've got carbon 1, 2, 3, 4, and 5. This 5th carbon is closest to this end of the molecule, closest to the attachment to the next nucleotide, and so we call this end the 5' end. On the opposite side, we look at the last nucleotide and its sugar. We count the carbons. We've got carbon 1, 2, 3. This 3rd carbon is going to be connected to the next nucleotide in line down here, and so we call this end the 3' end. All right, so a couple of things to review and maybe reintroduce. Nucleotides are linked together to build a polynucleotide. Remember, we're going to be linking the sugar of one nucleotide to the phosphate of another. We create a covalent bond to stitch these 2 monomers together. We have to give a special name to that covalent bond. We call it a phosphodiester linkage. It's a mouthful, right? Really, really long term there. Phosphodiester linkage. And that phosphodiester linkage, multiple phosphodiester linkages create that sugar phosphate backbone that we see in the polynucleotide, with the nitrogenous bases kind of hanging off to one side as appendages. And then really what allows DNA and RNA to store genetic information is that those nitrogenous bases that are hanging off to one side, their order, right? Where you have the A's, the T's, the C's, the G's, or in RNA, the U's, it matters, right? The order will determine, the order of the nucleotides or the nitrogenous bases will determine the sequence of amino acids that the cell will use to build a polypeptide. And so the sequence of the bases along the DNA or the RNA polymer is unique for every gene that's encoded in the genome. Let's talk a little bit about structure. Now, structure of nucleic acids is different when you're comparing DNA to RNA. So let's tackle DNA first. DNA molecules under normal conditions always exist as two polynucleotides that spiral around each other, forming what is called a double helix, what you see here in this figure. The interesting thing about these two polynucleotides that come together to form that DNA molecule is that they run in opposite directions. We call this anti-parallel, meaning that the two strands are parallel to each other, but you don't have both heads and both tails opposing each other. You actually have it flipped. So you have the head of one matching up with the tail of the other, and vice versa on the other side. And so you can see that here, right? The 5' end of one polynucleotide matches up with the 3' end of the other. So they are anti-parallel. One long DNA molecule, and this is only part of a DNA molecule, has many genes. It can have hundreds to thousands of different genes just on that one DNA molecule. So it holds a lot of information. And then when you have those two polynucleotides spiraling around each other, you also, to hold that structure in place, have complementary base pairing between the bases. So I like to think of DNA kind of like a rope ladder, right? So the rope on either side, that's like the sugar phosphate backbone, and then the nitrogenous bases come together in the middle to form the rungs of the ladder. A's are always going to pair with T's, there's no exception here, and then G's always pair with C's in DNA as well. And so you can see that here in the little blow up. The nitrogenous bases are held together using hydrogen bonds, which you can see here. This particular picture, though, is very deceiving because there are actually 3 hydrogen bonds that form between C's and G's, and only 2 that form between A's and T's. This is only just showing 1 for each. So remember, 3 hydrogen bonds between C's and G's, and 2 between A's and T's. All right, so RNA structure is very different, where DNA is always double helix, double helix, double helix. Doesn't matter whether you're talking about prokaryotic cells or eukaryotic cells or skin cells or protists, always double helix for the DNA. RNA molecules are much more variable in terms of their shape. There are different kinds of RNA molecules, and they'll have different folding, different shapes to them. Also, in contrast to DNA, RNA is meant to be single-stranded. And so this is an example of a special kind of RNA called a tRNA, and this is just one strand, one polynucleotide strand. Starts here, kind of loops around, goes down here, and then comes back. There's the other end. And you'll notice that there are certain places where the RNA flips back on itself, so it's anti-parallel to its own strand, and you can have some base pairing between the same RNA molecule parts to kind of hold that folding, that structure of the RNA together. And again, you have the same base pairing rules. C's are always going to pair with G's, but remember that RNA doesn't have any thymine, it uses uracil instead, so the A's are going to pair with U's in RNA instead of the T's like what you have in the DNA. Okay, so the last part here of the nucleic acid section is an animation. This animation compares DNA and RNA and goes through the structure and all that good stuff. This animation is found in Canvas for you guys, so you are welcome to watch that at your leisure.